Welcome back to Pharma Dermatology. Today I would like to talk about the immunology of the skin. So let's talk about the basis of the immunology. As you all know, the immune system is protecting our body from the invading viruses or microorganisms from the outside environment, and skin is one of the major immune organs. And the three functions of the immune organs or immune system are to distinguish between the served and the non-served antigen by recognizing the invading organisms or those antigens by immune receptors, and exclude the non-served antigens by producing information of the antibodies, and finally leaving the immunological memory. These functions are performed by the bone marrow-derived lymphocytes and antigen-presenting cells, such as macrophage and the B cells. This is an illustration about the myelogenous cells composing immune system. The hematopoietic stem cells are first differentiated into common lymphoid progenitor cells and the common myeloid progenitor cells. And the common lymphoid progenitor cells further differentiate into B cells, T cells, and natural killer cells. And the B cells can transform into plasma cells if necessary, and the T cells are further differentiated into cytotoxic T cells and the helper T cells. And the common myeloid progenitor cells have two main components, which are the granulocytes, including neutrophils, eosinophils, and basophils, and the macrophage. So in this immune reaction or immunity, we talk a lot about the antigen or antibody. So what are they? I would like to talk about the basis a little about antigen or antibody. So antigen is a protein of foreign substance, which can be bacteria, virus, or the transplanted tissue, or certain proteins recognized as non-served by our immune system. And so antibody is the immunoglobulin produced by B lymphocytes that is able to react against infectious invading organisms or pathogenic proteins, which is called antigen, inhibit the infection and neutralize the protein toxicity. And this antibody is a Y-shaped structure, and it has different regions for their respective characteristic, like FAB and FC fragments, and heavy chain and light chain. Here heavy chain and light chain, and variable region and the constant region. So FAB is the antigen binding fragment, which is recognizing antigen, and FC is a crystallizing fragment that reacts with other immune cells like macrophage and work with the complement system for immune activation. And the heavy chain is the inner Y-shaped structure. It has long peptides, and light chain is the outer segment with shorter peptides. And the variable region is the outermost portion of the heavy and light chain in the FAB segment, and is responsible for the binding of a variety of antigen. And the constant region defines the different isotopes of the immunoglobulin, which can be classified as GAMED in descending orders of concentration. Here I describe GAMED. So here in the variable region, the epitope is the antigenic structure that is responsible for the function and of the full potential of the antigen. And a paratope is the present on the variable region of the antibody FAB binding fragment site, and the antigenic the most antigenic portion, which is epitope portion, reacts with the paratope for the high affinity immune response. And so let's talk about the different types of antibodies. And so GAMED in descending orders of concentration, and it has its own structure, like IgG, IgE, and IgD. They are monomer, and IgA is the dimer structure, and IgM is the biggest and pentamer structure. So IgG is the most numerous immunoglobulin of all the immunoglobulins, and it has a function for humoral immune reaction. And IgA is abundant in the mucosa or mucus secretion, and it prevents the pathogen invasion. And IgM is release a very early stage of infection, which is responsible for the primary response, and it precedes IgG protection, and it strongly activates the complement system and responsible for the immune memory. And IgE is the binding to IgE receptor on mast cell and basophil, and it is responsible for the type 1 allergy and dermatology disease like atopic dermatitis. These serum IgE levels are very much increased, and it has an antiparasitic action. And IgD is a part of the B cell receptor, and it serves as a receptor for the antigen. So here I just added a little about hapten. So what is hapten? Hapten is not like antigen. It is non-protein and incomplete antigen. It has low molecular weight. It can be carbohydrate, fat, or some organic compounds, and it's attached to the carrier protein to be complete antigenic structure to elicit the immune response. For example, like we can say hapten in contact dermatitis or drug-induced SLE. They are non-protein hapten molecule. So let's talk about the immunity. Immunity is the state of being immune, which is the tolerance or adaptation of the immune system by protection of the invading microorganisms or counteracting the immune response by producing antibody formation. And there are two main types of immunity, which are the innate immunity and the acquired immunity. Some said there are three types, which is another passive immunity, which is left behind in this topic. And the innate immunity recognizes the invading microorganism and activates neutrophil, macrophage, and natural killer cells and complements. And it is a quick response to attack the invading microorganisms such as bacteria, and the LPS, which is which is found on the bacterial cell wall, joined with the toll-like receptor 4 on the macrophage surface, and sequentially triggers the acquired immune system, lymphocytes and the macrophage, and those cells such as macrophage, phagocytose, and excluded those invading bacteria, but the response is lower than the acquired immunity. So acquired immunity is an antigen-specific immune system, and it acts through the MHC molecules, major histocompatibility, molecule 1 and 2, and reacts and adapts to the various foreign subjects. This is a note about the difference characteristic between the innate and acquired immunity that can make you understand a little bit more easier. So the innate immunity is present at birth, whereas the acquired immunity, it happens after exposure to foreign antigens. And the antigen specificity is non-specific in the innate immunity, but for acquired immunity, it is specific to antigen after exposure. And the immune response is taking place rapidly within hours, but the reaction is less potent in the innate immune arms. But in the acquired arm, it is the response is slower. Usually, it takes about days, and which is less potent in contrast. And the immunological memory is absent in the innate arms, but the acquired arm, it is immunological memory is present. So in the cellular part, the innate immunity mainly involved by the phagocytes such as macrophages and neutrophil cells and the dendritic cells, but the humoral part is usually by the lymphocytes, mainly by the lymphocytes. And for the humoral immune response, and in the innate immune system is usually through the complement system, but the acquired immunity usually use the antibodies for the humoral immune response. And the physiological barriers in the innate immunity are skin and mucosal membrane, and in acquired immunity, lymph nodes and the spleen and the mucosal associated lymphoid tissues are the normal physiological barriers. Thank you for your attention. If you like this